So our next set of talks, we're moving to the Southern Ocean, staying in the Southern Ocean, and we'll be focusing on iron in the Southern Ocean. Yep.
Okay. Great. Hi, everyone. So I'm going to be talking about something a little bit different than I think a lot of people here. Oh, sorry. I guess I was a little too far away. I'm going to be talking about something a little bit different than I think a lot of people have been talking about here. And I'm going to be looking at subcellular proteomics for determining iron-limited remodeling of plastids and diatoms. So my name is Christopher Gomes. I'm at the University of Rhode Island. I'm in Dr. Bethany Jenkins' lab. And as I said, we're investigating diatoms, which are important primary producers, but their rates of primary production as well as their growth rates are significantly limited by low iron levels. And in the marine ecosystem, large regions of the oceans are predicted to be iron limited. So in order to try to investigate how these marine organisms are being affected by this iron limitation, we performed in-lab culturing experiments using the model diatom species Thalassiosauria pseudonana to try to understand how iron limitation affects protein expression within the chloroplast, which is the photosynthetic organelle. So to do this, we grew them under high iron and low iron conditions, and this is our pipeline with which we did this. But really what it boils down to is we dissolve the silica frustule, which is that thick glass cell wall that surrounds the diatom, with ammonium fluoride. We gently lyse the cell to pop the plasma membrane surrounding it, and then we enriched for intact chloroplasts. And we sent those off for protein sequencing, and this gives us an idea of the entire protein content of that organelle, which we refer to as the proteome. And so using the protein content between both high iron and low iron conditions, we can make an assessment of what is going on in terms of regulation. So if we look at the protein content, we had a total of 929 proteins between both conditions. This is the distribution of those proteins. We have 596 that are shared between both, 212 that are unique to iron limitation, 121 that's unique to iron replete conditions. And what we observed is alterations in rates of photosystem protein recycling, as well as regulation of light harvesting complex proteins and non-photochemical quenching. So what this shows us is that under iron limitation, these organisms are exhibiting high light stress, which is a result of the iron limitation, and this reduces their photosynthetic efficiency. So they're responding to that with these processes, and we also see a reduction of the proteins involved in carbon fixation in response to this. So this has implications for the marine food web, as there is a reduction in the amount of carbon that's being fixed by these organisms under iron limitation. That's it.